A frozen embryo is drifting in orbit. It has two cells. It is smaller than a grain of sand. It sits sealed inside a palm-sized incubator. That incubator is bolted to a wall on the International Space Station. Right now, this is humanity's closest attempt at a pregnancy beyond Earth. It is not attached to a mother. It is simply waiting, frozen, to find out if it can grow further. Here's the strange part. NASA's own experts say the human body works fine up there. Space medicine specialist Jennifer Fogarty at Baylor College of Medicine put it plainly. Anatomically and biologically, she says, there are no known impediments to human conception in space. And yet NASA's official policy forbids it outright. Not as an unspoken taboo, not as a joke astronauts trade in orbit, as policy. NASA is terrified of pregnancy in space not for moral reasons. It is terrified because no embryo has proven it can grow into a healthy human, not without Earth's gravity, not without its radiation shield. So if the body works fine, what is NASA afraid of? Something invisible. Astronauts have joked about zero-G arousal for decades. The internet asks the same question constantly. Can you actually get pregnant in space? But almost nobody asks the harder question. If nothing anatomically blocks it, why has no agency ever tested it in orbit? What could go wrong that makes ignorance the safer choice? The answer isn't about ethics committees. It's about physics. Start with the environment, just the raw physics. Aboard the ISS, gravity drops to about 1/millionth of Earth's pull. Scientists write that as 10 to the -6th G. Fluids stop settling. Cells lose all sense of up. Nothing pushes bone or muscle to build strength. At the same time, astronauts sit outside the Van Allen belts. Those belts are Earth's own radiation shield, and up there they don't reach. Astronauts absorb galactic cosmic rays constantly, roughly 0.65 to 1.8 millisieverts a day. NASA's Curiosity rover measured this directly, using its rad detector on the way to Mars. A single round trip adds up to about 0.66 sieverts total. That's a full-body dose, absorbed slowly over months. Compare that to 2 or 3 millisieverts a year on Earth. A chest X-ray gives you about 0.1 millisievert. Now imagine thousands of tiny versions of that striking cells every single day. That's roughly 200 times more radiation just from getting there. There is no atmosphere up there to absorb the particles. There is no magnetic field either. Two invisible forces. No gravity, no shield. Everything that follows comes from those two facts hitting a developing embryo. Let's test the first assumption people carry. Maybe conception itself just doesn't work up there. Maybe sperm can't find an egg without gravity's help. In 2000, researcher Kojima ran simulated microgravity fertilization experiments on mammalian cells. The result was almost boring. Fertilization proceeded essentially normally. Sperm meeting egg was never the actual problem. The mechanics of union don't need gravity's help. Whatever NASA fears, it isn't that first step. So the failure happens later, somewhere specific. And it does, almost immediately. In 1998, the space shuttle mission STS-80 carried a real test. Researchers Schenker and Forkheim at Israel's Aerospace Medicine Institute sent up mouse embryos. There were 49 two-cell embryos in total. Every one was exposed to genuine microgravity, not a simulation. None of them developed any further. Not a single one. 49 attempts, 49 dead ends. That result wasn't an isolated fluke. China's SJ-8 satellite mission repeated it in 2008. Two separate space programs, years apart, hit the same wall. For years, this became the accepted scientific position. Real microgravity was assumed lethal to early mammalian embryos. Full stop. No exceptions. Case closed. Then, in October 2023, that broke completely. Molecular biologist Terahiko Wakayama led the break. His team at the University of Yamanashi ran something called the Space Embryo Project. First, they froze two-cell mouse embryos here on Earth. Then, a cargo capsule carried them up to the ISS. Once aboard, astronauts thawed the embryos inside a compact automated device. That device could culture, photograph, and preserve embryos without a human hand touching them. A tiny built-in camera tracked each embryo's shape hour by hour. No crew member had to intervene at any stage. They cultured those embryos for 4 days in orbit. Some of the embryos actually survived. The survivors developed into blastocysts successfully. Those blastocysts showed normal cell counts and a normal inner cell mass. The inner cell mass matters because it becomes the actual fetus later. Their gene expression closely matched Earth-based control embryos, at least at that early stage. This was published in the journal iScience. It was the first proof mammalian embryos could pass the 2-cell stage in real microgravity. But notice the catch, because there always is one. Survival rates stayed lower than on Earth. Development stopped completely at the blastocyst stage. Nobody has tested whether implantation would follow. Progress then, but a fragile kind of progress. Even the survivors carry hidden damage. China's SJ-10 satellite mission looked closer in 2020. Researcher Shen Lei examined mouse blastocysts grown in orbit at the genetic level. The team found DNA hypomethylation at over 6,130 sites. Methylation is one of the switches that controls which genes turn on. Think of it as tiny chemical tags sitting on the DNA strand. Those tags tell a cell which instructions to read and which to ignore. Disrupt enough of those tags, and development can drift quietly off course. They also found elevated DNA damage throughout every sample. This traces back to microgravity-driven oxidative stress. A stress pathway called SAPK-JNK gets triggered in orbit. That pathway interferes with how genes switch on and off. The embryo can look intact on the outside. Underneath, its genetic instructions are already being rewritten. Now assume implantation worked, just hypothetically. The story still isn't over. A growing fetus needs mechanical resistance to build bone correctly. Physician Slobodan Sekulic proposed this back in 2005. Bones and muscles develop through what's called gravitational loading. That's the constant push and pull Earth's gravity applies. Every kick, every twist against gravity is quietly building tissue. On Earth, a fetus practices against gravity long before it's born. In orbit, that resistance simply disappears. A 2024 NASA-funded mouse study confirmed the mechanism directly. It focused specifically on long bone mineralization before birth. Without resistance, fetal bones fail to mineralize properly. Minerals like calcium never get deposited the way they should. Postural muscles never build the strength they'd need on Earth. On Earth, a fetus is training for gravity before it's even born. In orbit, there's nothing to train against. The exact thing a growing skeleton depends on simply isn't there. That's the mechanical story. But a second, independent line of evidence exists too. This one comes from living animals, not cell cultures. NASA-funded researcher April Ronka flew pregnant rats on real shuttle missions. After birth, the pups showed clear vestibular deficits. Their balance systems were measurably disrupted. They also showed delayed motor development afterward. This wasn't a theory about cells in a dish. This was a measurable outcome in a living, breathing animal. An animal born after its mother had actually flown in space. Two entirely different fields, cell biology and behavioral physiology, arrived at the same warning. A third line of evidence comes from pure physics, not biology at all. The Southwest Research Institute analyzed data from Curiosity's RAD detector. They confirmed cosmic ray dose rates near 1.8 millisieverts daily during transit. This number doesn't depend on cell biology or gravity at all. It doesn't care whether an embryo is fragile or resilient. Any embryo conceived during a Mars transit gets irradiated continuously. That's simple dosimetry. It happens no matter what else goes right at the cellular level. Here's where they stop acting alone. Together now. Radiation causes DNA double-strand breaks directly. Microgravity independently raises reactive oxygen species inside cells. Reactive oxygen species are unstable molecules that damage nearby structures. On their own, repair systems can often manage one problem at a time. Together, these two stressors overwhelm those same repair pathways. Cells with a defective repair gene called RAD9 show far worse break damage in microgravity. Recent reviews confirm this interaction directly. The two factors don't simply add together, they interact and the damage compounds. A cell that would normally survive one hit now faces two at once. This compounding, unrepaired risk is the concrete reason experts call NASA's rule an outright ban. Not every creature struggles here. Not all of them. Nematode worms have completed full life cycles aboard the ISS, generation after generation born and matured entirely in orbit. Certain insects have shown at least limited reproductive success, too. That contrast matters enormously here. It isn't life itself struggling up there. It's specifically mammalian development that keeps hitting a wall. Worms don't need gravitational loading to build a skeleton. Mammals do every single time, without exception. Step back now, look at the full shape. Fertilization works fine in microgravity, exactly as Kojima showed in 2000. But the very next stage, cell division past two cells, failed completely in 1998. It failed again on China's SJ-8 mission a decade later. That failure held as consensus until October 2023. Then Wakayama's space embryo project proved partial survival was possible, even though survivors carried hidden genetic damage per the SJ-10 findings. Assuming implantation somehow worked, a second problem waits regardless. No gravitational loading means bone and muscle can't mineralize properly. Living proof of disruption already exists in Ronka's rat studies. And underneath every biological finding sits one more fact. Cosmic radiation strikes regardless of what happens at the cellular level. Combine that radiation with microgravity's oxidative stress, cell repair systems get overwhelmed from two directions at once. Not a moral objection, that's an engineering problem written in the language of cell biology. Zoom out now past the ISS and every satellite mission mentioned. For 4 billion years, every organism here has built a body one specific way. It built inside one gravitational field. It built beneath one magnetic shield. That was never a footnote. It was the actual precondition for life as we know it. Remove gravity, and cells lose their directional cues entirely. They can no longer divide correctly or build tissue layers in order. Remove the shield, and radiation gets a straight line through. It strikes the genetic blueprint every single cell is copying. Picture a construction site, slowly tumbling in zero gravity. The bricklayers can no longer tell which way is down. Walls go up crooked, or they don't go up at all. Now picture an X-ray machine running just outside that site. It quietly corrupts the blueprints the crew depends on. Neither problem alone is necessarily fatal. A tumbling site might still produce something usable, and some radiation exposure might not matter much either. But combine both failures on the same site at once, nobody can guarantee the structure holds. That's exactly what NASA's policy answers. Precisely that. So gather it all together, and the real story finally lines up. Gravity isn't background scenery for a developing embryo. It's an active construction tool, one mammalian biology never learned to build without. Radiation shielding isn't a bonus feature of living on Earth. It's the reason your DNA has stayed readable for generations. Take away either one, and you get real uncertainty. Take away both together, and you get exactly the compounding damage this investigation just traced. It started with a 1998 shuttle failure. 49 embryos, 0 survivors. It moved through a 2023 partial success, fragile blastocysts, no implantation tested. It ran through the genetic fingerprints found in 2020, thousands of altered switches. It carried through rat pups with disrupted balance and bones that never mineralized right. All of it converges on one unresolved risk. That's the exact risk NASA has quietly guarded against all along. NASA's ban isn't caution for caution's sake. It's an admission, plain and simple. Humanity doesn't yet know how to build a person outside that cradle. Earth's gravity has always done the job for us. So has its shield, silently, every single day, for every creature ever born here. Which brings us back to that frozen embryo. It's still sealed inside its palm-sized incubator. It's still orbiting Earth every 90 minutes. It survived longer than almost anyone expected. It still hasn't been allowed to finish the journey. Somewhere above your head right now, it's still waiting to find out if it can.